to record until after I pray. But I already set it to record. So pray with me. Father, thank you for just taking care of our sin problem. Father, I thank you that you know us better than anybody alive. You know us better even than ourselves. And you know our selfish thoughts and ambitions and desires, and how even as your children, we oftentimes place our own authority above your own. And Father, you love us in spite of all that. And you not only do you love us, but you want us to know you and to love you more. And Father, I thank you for that, and I thank you for the hope that that provides for our eternity, but even the joy that it provides us now, and knowing that we get to walk with you and walk alongside you and have your Spirit literally dwelling within us, guiding us and making intercession for us. Father, I ask that all of this would drive us to work hard at whatever we do. Work hard in our classes, work hard at our jobs, work hard as friends in a relationship, work hard as brothers and sisters and sons and daughters. And Father, when we get this opportunity as spouses and parents, and I just pray that our drive would be something that would show people your grace and your love. And we ask this in the name of Jesus. Amen. All right, so we've mentioned already the major lipids at work in cell membranes, and what are they? What are the major lipids at work in cell membranes? Phospholipids and sterols, specifically cholesterol, but sterols. Both of those have both a polar and a non-polar region, so they are both water-loving and water-fearing. But there are other molecules, other biomolecule types that aren't lipids that play a very important role in cell membranes. Proteins being the major one. And here are some of the things that proteins do inside of cell membranes. One is working as transporters. Polar materials do not move easily through a lipid bilayer, right? And so they need to be transported into the cell. And what we're talking about here is not moving something against its concentration gradient, but just allowing it to move the direction it wants to move, from outside the cell to inside the cell, or from inside to outside if that's the way the concentration gradient is going to take it. But these transporters function to allow those materials to move by diffusion. Identifiers. Identifiers are proteins that basically announce to the external environment what that cell is and where that cell should be. And so they are identifiers that will declare your cell as being your cell, but they will also declare the cell as being a specific type of cell: red blood cell, liver cell, neuron. All of those have specific identifiers on their surface to declare what type of cell that they are. Receptors. And so receptors are on the surface like identifiers, but rather than declaring what that cell is and where that cell ought to be, these are cells that function to bind material in the environment to elicit some response from the cell or to bring that material inside the cell. And then finally, proteins function in adhesions. We saw this with desmosomes and adherens junctions. We saw it with tight junctions and even gap junctions. These proteins functioning in the membrane to hold cells together, either to keep them from being torn apart because of extreme forces, or to keep materials from moving in between the membranes, or to allow material to move from one cytosol to another. And then the sugars of glycolipids and glycoproteins form a coat around the cell called the glycocalyx, contributing to that extracellular matrix. Bless you. And so you have sugars that are on top and that are part of lipids, and you have sugars that are part of proteins functioning in cell membranes. Okay, so let's think about this. If there is a protein that spans all the way through the membrane, and here's what our membrane looks like. here's what our membrane looks like, right? We've got our lipid bilayer, and then we've got, some sterols in there as well. Whatever, right? We've got some sterols in there as well. So here we have a polar region, and so these head groups are polar. They interact well with water. The inside here, these are non-polar. And so if you're going to have a protein that's going to span through this lipid bilayer, what should be the characteristic chemistry of the portion of the protein that's in this section? How would you characterize, or what would you predict about its chemistry? Polar or non-polar? Non-polar. And so what you'll find is the residues, the amino acids that make up this portion of the protein, are non-polar. And the amino acids that make up this portion of the protein are polar. And so that the protein has predictable chemistry, but also almost is destined to end up inside of that membrane. If the phospholipids are going to self-assemble in water, proteins that are built like this, that have exposed non-polar residues and exposed polar residues, are going to spontaneously end up in membranes as well. Right? Right. This is good stuff, even if, you can't see it yet. And so this is a typical membrane protein. Portions that extend outside of the cell, portions that extend inside of the cell, and then portions that are embedded in the membrane. And again, if you looked at the amino acids for the portion that extends through the membrane, what you would find is that those are non-polar amino acids. They have R groups. Remember the amino acid, typical amino acid looks like this. Typical amino acid looks like this, right? And this R is what's different from one amino acid to the next. This R is what determines whether that amino acid is non-polar or whether it's polar. Okay, and so what you'll find is the amino acids in this section have non-polar R groups. And so they interact well with the fatty acid tails. And so here's just not what the protein actually looks like, but just a cartoon of what that previous protein would do, where it forms this channel on the inside. And so on the outside here, you have non-polar residues interacting with the lipid fatty acid tails, right? But on the inside of this channel, you have polar R groups that would allow material to easily move through here. They're interacting with water out here. They can interact with the polar R groups on the inside of this channel, and then can interact with the water molecules here. And so now it becomes a nice, easy way for them to just experience the same chemistry all the way through. And then so what's driving them to move through then if it's not trying to escape bad interactions? The diffusion, right? Moving down their concentration gradient. And so now it allows diffusion to be what causes them to move and not trying to avoid negative interactions. Right? Yes. Okay. Next question. What is the structure of membrane? So if we were to, attach a name to describe the way in which these behave, or if we were to, come up with an idea of just what are these membranes like, what would it be? And so the dominant model of cell membranes is called the fluid mosaic model. And what is a fluid? A fluid is anything that is not a solid. Liquids are fluids, gases are fluids. Anything that is not a solid is a fluid. Okay? And so this idea here, this fluid model, it's reminding you that this membrane is not fully solidified. Right? We talked about last time, what are the consequences of having a solid membrane? And those aren't good consequences, right? So it's fluid. It's able to move around, right? Saw this going on when I asked what is a fluid. And that's exactly what's going on with the membrane. And then what is a mosaic? It's something made up of a lot of different parts, right? And so you can see like a table that's a mosaic. And so from far away, it's like, wow, it's a beautiful owl on that table. And then you get up close to it and you realize it's all these different types of glasses all mixed together. And from up close, it looks kind of chaotic, but from far away, it gives you this, nice image. And so a mosaic is just something made up of a lot of different types of materials. And that's also true of the cell membrane, because you've got phospholipids, you've got sterols, you've got proteins, you have glycolipids, glycoproteins, all of these different materials making up the cell membrane. So it's fluid, right? And it's a mosaic. Yeah.
Do different types of cells have different levels of optimum fluid?
Yes, absolutely. Yep, absolutely. And it tends to be dependent upon the optimal temperature of that species. So if a species operates at an optimal temperature of, 40 degrees Celsius, which is pretty hot, but there are plenty of organisms that live in hot pools. Their membranes are more fluid than, say, our membranes are. Yeah, there is an optimal fluidity. So phospholipids move freely and fluidly. Phospholipids can move from here over to here. They can move from here over to this side of the lipid bilayer. They can move from here over to here. They're free to move around within that matrix as long as they don't expose their fatty acid tails to the water, to the polar materials. Yeah, so they move really freely within layers, and then to move from one layer to another, it still happens, but it's not quite as flexible. It's not quite as flexible. All right. And so now we need to talk a little bit about fatty acids. And so fatty acids, you have saturated fatty acids and you have unsaturated fatty acids. Okay? And so a saturated fatty acid looks like this. Okay? Every vertex, every point is a carbon. Okay? So every point is a carbon. And then anywhere where there's nothing written, you can assume there's a hydrogen. So this carbon wants to make how many bonds? 4, right? That's what carbon likes to do. Well, it's making one bond to this carbon here, right? This is a carbon, 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 this is a carbon here, this is a carbon here, right? So this carbon has one bond with this one. It has one bond with this one. That's only 2 bonds, right? So you can assume that it's also got this going on, that it's bound with 2 hydrogens as well. Okay? If it is saturated, it has no carbon-carbon double bonds. Okay? Another way of thinking about that, it has as many hydrogens as you can possibly hold in that hydrocarbon. And remember, fatty acids, they're just hydrocarbons. They're just carbon and hydrogen, okay? So it has, actually that's not even true, because at the end of the fatty acids you've got a carboxyl group. But anyways, for a long stretch of it, it's just carbons and hydrogens, all right? If it's saturated, it has no carbon-carbon double bonds, and it has as many hydrogens as it can possibly hold. And that's why we call it saturated. It's saturated with hydrogen. Unsaturated, these have carbon-carbon double bonds. And so you'd have something that would look like this. Okay, here's a carbon-carbon double bond. So this carbon has 2 bonds with this carbon, right? And 1 bond with this carbon. How many bonds is that total?
3.
3, and so then you would assume that it's got 1 bond to hydrogen. Well, that's 1 less than it ought to if it were saturated, right? So it is not saturated with hydrogens. Instead, it is unsaturated because it has this carbon-carbon double bond. Okay, unsaturated fatty acids, they've got a 3-dimensional, these are all 3-dimensional objects, but you basically have 2 options for this carbon-carbon double bond. You could have this, where the 2 big bulky groups are on opposite sides of that carbon-carbon double bond. This is called trans. This is a trans fatty acid or a trans fat. Okay, have you heard that before? Trans fats. Okay, or it can look like this. Let's try to get this the same. 1, 2, 3. 1, 2, 3. Okay. Or it can be this, and this is cis. Okay? This is a cis unsaturated fat. The big bulky groups are on the same side of that double bond. Okay? What this does, having a shape like this, makes a bend in your fatty acid. Do you see that? It bends. And so instead of sitting straight like this saturated fatty acid does, it's bent. And so what it makes it is where it's really hard to pack these together. It's really hard to pack these unsaturated fats close together. And so what it makes is, it makes it to where these unsaturated fatty acids tend to remain fluid even at low temperatures, because you can't pack them close enough together to form a solid. Okay? And so this is what actually helps to maintain fluidity, is you can't really pack them real tightly here because they're so bulky. You know, they're bent and just take up a lot of space, whereas this, you know, is straight and you can pack these things a lot more tightly together. And so this is why animal fats like butter or beef fat or bacon fat, the best animal fat there is, they have a lot of this saturated fat. And so they can pack really tightly together and they're solid at room temperature. But vegetable fats have unsaturated fats, and they don't pack as tightly together, and they're liquid at room temperature. Yeah, Joe.
Okay, so because we remember this like in chemistry last semester, like a double bond tightens it up so that it can only rotate, so that it can't like rotate on the axis. So not understanding how like the carbon double bond, like
what it doesn't fluctuate between the two.
Okay.
Right? It doesn't like at 1 minute it's trans, at the next minute it's cis, and then it goes back to trans, and then it goes back to cis. Okay. No, it's locked. It's locked in that perspective. Yeah, absolutely. Those double bonds, those pi orbitals, where those electrons are, do reduce torsional flexibility. Okay. And so if it's trans, it's stuck that way. If it's cis, it's stuck that way. Okay. To I don't know, address your concern.
Okay, and then like the saturated one, like is there like flexible movement within that line, or is that like a locked, the saturated fat?
Well, I mean, all solids vibrate.
Yeah.
I mean, there's movement even within solid material, right? There's some vibration, and so that I mean, these are free to move. Now, at low temperatures, there's less kinetic energy, right? Temperature is just the average amount of kinetic energy. So if they're not moving as much, you can pack them more tightly together. But if they're moving a whole bunch because they're warmed up, they're not going to be packed together anyways. They're moving around, and the vibrations are just keeping them loose and keeping them from solidifying anyways.
Okay.
Yeah.
So do the carbons in the saturated fat move independently of each other or all together?
Both. They have some flexibility to move in all dimensions. And so the bond length, you know, when you give like a bond length, have you all talked about bond length in Gen Chem?
Yeah.
Okay, well, when you talk about bond length, it's average bond length. It's what is the average distance between these two atoms. But that bond length shifts. Sometimes they're closer together, sometimes they're further apart. And the more energy that's in this, the more flexibility there is. And that's why eventually it gets to a point where these bonds start to break and things start to liquefy. Is because those materials are moving around. As you heat them up, they have more kinetic energy. They're moving around. Eventually, they get to a point where they're moving fast enough that they can completely break apart. And then your solid is now a liquid. Or your liquid is now a gas if they're completely kind of independent of influence. Not completely independent, but much more. So unsaturated fatty acids help to maintain fluidity at low temperatures, keep it from solidifying. And then cholesterols help to keep it from getting too fluid at high temperatures, because phospholipids have a lot more freedom to move around than cholesterols do. Cholesterols, they don't have these fatty acids. Instead, they've got these big clunky rings, right, inside. So they're not as free to move around as these phospholipids are, and that helps to keep it from getting too fluid, right? Because it's like, yeah, it's fluid, but you wouldn't exactly call it a liquid. Like gas is a fluid. Glass is actually a fluid, but you wouldn't exactly call it a liquid, if that makes sense. Have you ever been in a really old house and looked at the windows and they're thicker at the bottom than they are at the top? It's because glass is a fluid. But you wouldn't call it a liquid, but it's a fluid. There's a great deal of freedom to move around. And so gravity pulls it down, and so in like old windows, they're thicker at the bottom than they are at the top. Check it out next time. There aren't any old houses in California. You got to go somewhere in the country where there are old houses. 300-year-old houses. David's neck of the woods. All right. So proteins mostly float around individually. They aren't. These non-polar interactions are not as strong as polar interactions. So like the proteins that are operating in here on the outside, they've got non-polar materials that don't repel this, but non-polar interactions aren't. They're not like holding hands and locked in place. They're just kind of coexisting. And so, some are actually, if you want to keep a protein attached and in place and not moving around freely, you have to anchor it to the cytoskeleton. And so a lot of proteins in the membrane can't move around. They need to stay on a particular portion of the cell. And this is especially true if the cell's locked in an extracellular matrix. Then you need protein receptors on one side. You have to anchor those proteins to the cytoskeleton, otherwise they'll float around like the phospholipids do. And so, you see this integral proteins, like these are embedded. Peripheral proteins do not penetrate the lipid bilayer at all, and instead are just attached to either the outside or the inside. And you see here attached by non-covalent bonds, so it's just they're polar, these are polar, they're attached primarily by hydrogen bonds interacting like water molecules do. And proteins are arranged asymmetrically, and I'll show you what that looks like. So the proteins on the inside of the lipid bilayer are different than the proteins that are on the outside, which makes sense, right? Because some of those proteins on the outside, they are identifiers, right? Declaring what that cell is and where it ought to be. You don't declare that on the inside of the cell, right? The cell knows who it is, right? It's the way of expressing itself to its surroundings. Okay, so here's a representation of kind of this mosaic. It's not really illustrating the fluidity, right? It's a cartoon. It's locked and it's locked. It's not really illustrating the fluidity, but it certainly illustrated the mosaic, right? That there are a lot of different components making up this cell membrane. So the cell membrane is not just the simple lipid bilayer with nothing else involved. It's a very fluid lipid bilayer with lots of other materials sprinkled in. Yeah.
Can you go back to the slide for like 3 seconds?
Yep. Yeah. Wow, we're already at 12:10. Gosh, you guys need a lecture break. Let's do this, and then we'll take a lecture break. All right, so here's the mosaic. Here, even representing in a cartoon, is representing the fluidity. And so here they built, they basically fused two cells together. You have a human cell with human peripheral proteins. You have a mouse, and this is just fusing the membranes, not the entire cells. If you tried to fuse the entire cells, weird things would happen. But anyways, you've got human proteins, you've got mouse proteins. You fuse them together, and so you end up with human proteins on the top, mouse proteins on the bottom. But you give it some time, 40 minutes, and now you get a mix, showing you that these proteins are moving around within this lipid bilayer. So this is demonstrating the fluidity of this membrane. All right? So we need to take a lecture break, after which we'll take our quiz. So use this time in the lecture break wisely to, maybe ask some people around you about any questions you might have on Chapter 5 as a whole in preparation for our quiz. So here's what I want you to do for our lecture break. I want you to give 3 benefits. You can still call them consequences. They're just positive consequences. But 3 consequences of a fluid membrane. 3 consequences of a fluid membrane. Try to emphasize positive consequences, like how that actually benefits the cell to have a fluid membrane. but provide 3 consequences of having a fluid membrane. Okay? And then as a group, decide somebody that's going to represent your ideas to the group at large, and take a couple of minutes to do this starting now.
I wasn't here on Monday. What group did you
You were here on Friday?
Yeah.
I mean, you could.
Okay.
Okay. Do you like that group?
Yeah. Okay.
Yeah.
We're talking about like this. Yeah.
So it should be bad.
Okay. That's like that'd be like a negative consequence. Oh yeah, but he said you could be. He said like a consequence. He's kind of using it.
There's 2 main ones, I think. It was like and then they also
So it allows, especially in like, amoebas, right?
yeah, so yes, the consequences of having
What's the question though? I do not blame you. I think that is a good consequence.
yeah, he said negative 2.
I was thinking like it allows it to adjust like its size, like for normal water. Yeah.
It's nice and warm.
Yeah.
It's already partially chewed.
Or even like talk about how I don't know, maybe I was reading this somewhere how like
The best thing is when your sibling
red blood cells they have to like
So you didn't actively seek it out and found you. All right. So what are some consequences of having a fluid membrane? What are some consequences of having a fluid membrane? All right, we'll start with this specific group. So we'll start here and we'll make our way counterclockwise. What do we got, Joey?
allows for movement.
Allows for movement of what?
Yes.
Okay.
It allows for movement of the cells, like you have like amoebas.
Yeah, I like that. Getting the entire cell. Amoebas with their what are called pseudopodia, of actually just ramming. Basically, what they do is they ram their cytoplasm against the membrane and get the entire membrane flowing in a particular direction. And it's awesome. And yeah, that is certainly a positive consequence of having a fluid membrane. Getting that whole thing, and you can even get it rolling. You'll see this with white blood cells. They'll just kind of roll the entire cell, just along something. And it's actually really cool. So like if a white blood cell is being recruited somewhere, it'll start to slow itself down by binding materials in the lining of the vessels. So it's just kind of rolling along or moving in the bloodstream, and then it like hits and just rolls and like binds things and slows itself down so that it can then move in through the capillaries. All right, what do we got here?
We talked about how it provides with help with flexibility.
Sure. Yeah, not just with movement, but also allowing the cell to change shape. Part of that is movement, but there's more than that as well. We'll talk in this very chapter about phagocytosis, endocytosis, exocytosis. Those don't require the cell to move, but certainly require some flexibility in the membrane to be able to, bring vesicles in and take vesicles out. Any thoughts back there, group 4?
We agreed with the first 2.
Okay.
But also kind of negative is it could, it's a little bit more sensitive, so it's able to be damaged more, potentially dry out.
Potentially, sure. I mean, the if it's going to act as a barrier, the warmer it is and the more freely materials are moving, the less inhibitory or inhibitive that barrier is, right? Group 3, we have some unique thoughts, ideas.
We were talking about kind of similarly the permeability of the membrane.
Okay.
So things can be taken in and
Yeah, that's a positive consequence, right? And so if you have materials that don't move well through a lipid bilayer, heating those lipids up actually allow, just by sheer velocity of materials, to get some things through that may not normally get through. And water's a big one. So water, even though it's polar, really polar, can get through cell membranes as long as they are fairly fluid. When it gets cold, water has a really hard time moving across membranes. Yeah. Group 2.
We kind of the same thing as what everyone
Okay.
said, but like it makes it able for material to be transported.
Yeah. Good. Yeah, so you've got your endomembrane system, right? And it's shuttling materials in vesicles from one structure to another. And then any material that needs to go to the plasma membrane is going to move from a vesicle, from typically the Golgi body, over and then fuse with the membrane. But what's nice is you can have a single roadway and everything ends up in the membrane in like the same place. But then because of fluidity, it's going to migrate around to where you want it, right? But you don't actually have to actively drive it there because it's going to get there. Yeah. Ladies, group 1.
A couple ideas we talked about were that it helps it stay alive and function in a larger range of temperatures.
Okay,
So
good.
Instead of like freezing immediately.
Right. One, solids, interesting things happen to solids as they heat up past a certain point, right? Think about a concrete, walkway, right? As you start to heat things up past a certain point, your crystalline structure can fracture. But having something being fluid allows for protection against that. Yeah, I like it.
We also said that since the proteins can move around, it can access the materials it needs regardless of where they come from.
Absolutely. Yep, absolutely. And that actually is a real big benefit with the endomembrane system. So real quickly, I just wanted to show you the research that showed that the outer membrane looks different from the inner membrane. So they froze these membranes and then cut the two layers in half. And the two layers are different from each other. They are not mirror images. There are some proteins that are functioning on one side and not really functioning on the other. So kindly put away everything except for your slip of paper. Does everybody have a slip of paper? I don't see anybody saying no. I see some people looking like they're a little disappointed that they do have a slip of paper. It might be nice to just kind of sit one out, right? Or have maybe somebody having two slip of papers, one with your name on it and one with their name on it. Just kind of do things for you. Okay. Oh, you have an extra one? All right, so make sure you put your name on the slip of paper. I know even though you're emailing it in, please make sure your name is on that slip of paper. It ensures that the proper quiz goes to the proper person. Alrighty, here we go. Name 2 types of proteins that help facilitate diffusion. You just very generally or specifically if you want. And then name 2 types of molecules that don't move freely across the plasma membrane. And in which type of solution will cells shrink? And I know we haven't gotten to this third one yet, but you've read the chapter in its entirety. So.
Isn't it question number 2 basically the same as the last time?
Except for the last time you only had to give 1.
Oh.
And now you have to give 2. and don't be afraid of seeing the same question appear on multiple quizzes. That will happen.
It's not because you just didn't make a new question.
Well, that would be interesting if it just was the same exact quiz twice. That probably will not happen. I got an email from Mackenzie, and I was like, "Where is she?" But she just walked out. All right, one more minute. Another 10 seconds or so. Wrap up your thoughts. Make sure your name is on the quiz. And snap a picture of it and send the email over.
Literally every prof in this class has done that during a break.
It's just, it's not how I pictured Noah in my mind. Pictured him with a much, much bigger beard. Much bigger.
I was picturing him with skinny jeans,
Yeah,
V-neck, big gold chain.
big gold chain with a clock on the end, like Flava Flav.
Yeah.
All right, so everybody sent it in. You've got your picture. You sent it in. We're good to go. Okay, so two types of proteins that help with facilitated diffusion. You could talk about transporters, channels. You could have said integral proteins. Any of those work as that. Two types of molecules that don't move freely. You could give two examples of a polar molecule if you wanted to, but anything polar and anything big isn't going to move very easily. Even if it's nonpolar but it's big, or if you wanted to give two polar molecules, that's fine. In which type of a solution will cells shrink? Hypertonic. When there's more solute surrounding the cell than there is inside of the cell. All right. Yes, sir.
You were mentioning before we had our quiz about so the higher temperature loosens up the membrane.
Yes.
So like when we get sick and you have a fever and your body heats up the temperature of the blood, is that any connection to what it's trying to accomplish with like cell membranes, or is that like?
Well, that's a consequence of that, of having your body temperature increase. The main thing it's trying to accomplish there is actually twofold. One is the pathogens are probably not as equipped as your body is to handle elevated temperatures. And so part of it's trying to, destroy pathogens. Another one is it just increases the rate at which you can move materials.
Okay.
Right, so you can recruit immune materials more quickly and more
So
efficiently.
it's not necessarily materials in and out of the cell, but materials around the body.
Right. But that is also going to be another consequence of it. As your body temperature increases, your membranes do become more fluid. Right, so materials are going to diffuse more easily. All right, so how does the membrane function? So just as a summary, what do the membranes do? I mean, they form boundaries. That one is fairly straightforward. They are permeable, that is, they're selectively permeable. They allow some materials to move across and not others. So they are a selective boundary. They can be enzymatic, and this is especially true when they have certain proteins exposed to the outside, proteins that can actually facilitate reactions, lowering the rate of the reaction, lowering the activation energy. Regulating transport of specific ions, and there are a couple of examples that I want to give you of actual real life channels that do this. So these are facilitating the diffusion of ions. They are not necessarily actively pumping ions. And so you have what's called the cystic fibrosis transmembrane conductance regulator. I don't know why the C is not part of this acronym, but it's not. So cystic fibrosis transmembrane conductance regulator allows chlorine to move down its concentration gradient. Oftentimes the cell will trap chlorine ions inside of the cell because it's a way in which it can rapidly kind of fix a charge issue. So when it opens up these channels and allows chlorine to move down its concentration gradient, which will be out of the cell, it allows for enough of a total concentration difference that water will follow the chlorine out of the cell. Okay? And this happens especially in tissues where you build up a mucus barrier to help to protect against pathogens, to protect against other things entering into the cells. And so you find this especially in the lungs, where you have a nice mucus layer facilitating gas exchange, but also protecting the bloodstream and lung cells from attack from pathogens. And so if these channels don't work properly, the cells can't get the chlorine to leave and can't get the water to follow, and so that mucus gets really, really thick. And it can start to inhibit proper lung function. And so you may recognize this disease, cystic fibrosis. So cystic fibrosis is not technically a lung disease. It is a malfunction channel. These do function in other places as well. But one of the major pathologies of that is inhibited lung function. And it's very unusual to have somebody with cystic fibrosis live past their late 20s without a lung transplant. Because their channels don't work properly. But if they get a lung transplant, somebody else's body built those lung cells, and so those lung cells should have properly functioning channels that can then open up, allow chlorine to leave and water to follow, and keep the mucus functioning well inside of that area. So this is just an example of how a membrane can regulate movement of specific ions, and usually it's by diffusion. So membranes can bind substances and then transport them across the membrane. We'll see several ways in which this can happen. They can recognize specific molecules, and this is especially in places where you have a great number of receptors. Conduct signals with electrical properties, and we see this especially in neurons. And in muscle cells, cells that are called excitatory. And finally, facilitate cell adhesion, so cells sticking together and then also facilitating cell-to-cell communication. So these are all functions carried out by the cell membrane. And so let's say hypothetically, on some hypothetical exam that's going to occur, I don't know, about 3 weeks from now, you were asked to provide 5 functions of the cell membrane and what carries out that, or what enables it to carry out that function. It probably wouldn't be 5 because that would be a big beefy question. But maybe if you were asked, I don't know, hypothetically, provide 3 functions of the cell membrane and what enables the membrane to carry out that function, I'd expect you to be able to answer that on this hypothetical assignment that may occur 3 weeks from now. Thank you, Charles. That was nice. I like the chuckle. Thanks. All right, so here's the channel, the cystic fibrosis transmembrane conductance regulator, allowing chlorine to move down its concentration gradient, sometimes even actively pumping chloride or chlorine ions out of the cell, and again, getting water to follow, helping to regulate the concentration of the mucus in wherever that tissue is. So again, cystic fibrosis is not technically a lung disease. Some of the pathology is in the lungs. It is a issue with a defective channel. All right. Any questions about that question or any of the previous questions that we've talked about, important questions or quiz questions?
There was actually treatment without doing an organ transplant.
Oh, there are. I mean, there are ways that you can treat the malfunctioning channel, but it still is very unusual to be able to do that indefinitely. Usually at some point, those treatments cease to work effectively. And it's usually sometime around the late 20s, early 30s that those treatments just stop working as well as the body, once you get into your late 20s, early 30s, you're well into the deterioration phase. There's basically only 2 phases. There's growth and development, and then there's deterioration. There's no real stasis. So by the time you're in your late 20s, early 30s, you're well on your way into that deterioration stage, and certain things just stop working as well. And so a lot of the efficacy of those treatments just cease. And that's why I said it's unusual. It's not unheard of. There are people that live past their late 20s, early 30s with cystic fibrosis and don't get a lung transplant, but it's rare. It's rare. All right, next set of important questions. How do materials move across the membrane? Here we'll talk about diffusion, talk about active transport, what specific proteins function to do this, to facilitate diffusion or to actively transport or pump materials. Why is osmosis so special? Why do we actually give it a separate name? Because osmosis is just diffusion of water. Why do we provide it a special name? And then how can we predict when and where osmosis will occur? And this is when we'll get into the third quiz question from today's quiz of when are cells going to shrink and how can you actually predict that? All right, so first question, how do materials move across the membrane? So, yeah, I mean, materials naturally diffuse. Anything is going to naturally diffuse. It's going to move from an area of high concentration to an area of low concentration. We call that moving down its concentration gradient. And then it basically, this is a type of passive transport. Passive meaning it doesn't require any energy. And passive meaning it doesn't require any kind of facilitation. It's just going to happen. So passive transport, it's just diffusion is taking place without being facilitated. Yeah.
Does facilitated mean like channel proteins?
Yes. Yep. Typically, although it could also be a transporter and not a channel protein. And so channel proteins just, again, they open it up and allow materials to move through. Transporters may actually grab hold of something and pull it through down its concentration gradient. That happens with insulin and glucose. Glucose is still moving down its concentration gradient, but insulin acts as a transporter, actually pulling it down its concentration gradient. So it could be a channel or it could be a transporter that actually physically binds it. So channels don't bind anything, they're just a tunnel to move through. But you have transporters that will actually physically bind materials and move them in. They're not pumping them, but they may physically bind them to move them in.
Would so like the diffusion of water across the cell membrane would be considered
Passive.
it's passive, but is it facilitated?
No. Well, partly. Some of it is through aquaporin, which is a protein channel, but some of it just happens through the membrane itself because of the membrane being fluid. So it's both. And that's why we'll talk about why is osmosis so special. Save that for that question. So yeah, diffusion is this net movement of materials down their concentration gradient. And so the reason why we care is this is a form of potential energy. So if you've got this concentration gradient set up where you have a large amount of something on one side of a membrane, a small amount of something on the other side, you have potential energy. You have energy that can be put to work, that can be used to actually do something. And so this can be simple or facilitated. If it's facilitated, it's helped by a protein. And in answer to Alyssa's question, that could be a protein channel, just allowing materials to move through. Or it could be an actual transporter that binds whatever that material is and allows it to come into the cell. Now, anything that you need to get to move against its concentration gradient is going to require energy. And so we call this active transport. And active meaning it requires energy. If you're going to move something against its concentration gradient, you're forcing it to move the opposite of the direction it wants to move. And so this requires net energy input, and that energy usually comes from breaking ATP, from hydrolyzing ATP, breaking one of those phosphate groups off and releasing the energy within that bond. Another thing just to keep in mind, again, membranes are selectively permeable, so polar molecules move through membranes very slowly and are inhibited by this large nonpolar region in the middle of this membrane. Charged ions or charged atoms, charged molecules are completely blocked. They will not move through membranes in most conditions. If you really heat those membranes up and make them really fluid, you could probably get ions to move, but under normal conditions, they will not move and will always need to be facilitated. Will always need to be facilitated. All right, the next slide is a table that does not appear in your text, but you have access to these slides, and it is now your table to use however you want. And it is a great just way to summarize all of these types of diffusion. So we've got passive, those not requiring energy, and then you have simple diffusion, and you have facilitated diffusion. And notice water is in both those categories. Water will move in by simple diffusion and will also move in by facilitated diffusion. And that's what makes water so special. Binding of transported substance, yes, in facilitated diffusion, unless it's just moving through a channel, but a lot of times you're binding it, and what's fueling it, concentration gradients, setting up that potential energy, which can then be moved and converted into kinetic energy or fueled by ATP hydrolysis. And then the last portion of that table just showing some additional characteristics of these transports. And so this table is not out of your text. You'll notice it's from a chapter 6 from a previous text, but it is now your table. And it is a great, great tool for you as you kind of work your way through facilitated, simple, and active transport. All right? Enjoy the rest of your Wednesday. Have fun in whatever classes you have left. If you have no classes left, have fun doing whatever you do on Wednesday afternoons. Yep. Sure. Yeah. They're on Canvas. If you click on the module tab, and then there'd be like module 1, module 2, module 3.
That'd be awesome.
On Friday, is there a way I can take the quiz early?
Yeah, that can work. Like, are, when would you want to do it?
Well,
sometime after dinner.
Just text me.
Probably like 7 or 8.
Just text me. Whatever time works.
Okay. What about if we just
Or we can
did
do
it
dinner, I don't
earlier
know,
in the
whatever your
class?
yeah.
So you still during our normal class time. That way you didn't have to.
Yeah.
Okay. Yeah, I'll make sure that
And then
happens.
I could just